Hi friends, today we are going to see the past year NEET questions for the chapter Microbes and Human Welfare, and today in this video we will cover part 2. Now let's begin with the first question here. Trichoderma hazardum has proved a useful microorganism for dash. So before we go into the answer directly, let's discuss what is this Trichoderma. This is actually belonging to the genus of fungi. So what is it? Fungi in the family of Hypocrisae. It is present in all soils where they are most prevalent culturable fungi. Many species in this genus can be characterized as opportunistic, avirulent plant symbionts. So this is an introduction about this. Now when we go particularly for Hazardum, this fungus, if you see, it is also used as a fungicide. What is it used as? A fungicide. It is used as a foliar application, seed treatment and soil treatment for the suppression of various diseases causing fungal pathogens. Okay, fine. Now we are asking how it is proved to be useful microorganism. The correct answer is option D, biological control over soil-borne plant pathogens. Trichoderma hazardum has proved to be a useful organism for the control. As I said, we can use it as in the soil or in other pathogens, especially the plants the pathogens that arise from the soil. Okay, they are called as soil-borne pathogens. Trichoderma hazardum is a fungus that is also used as a fungicide. It is used for the foliar application. So whatever I explained is to your right side. Okay, it's the same. Apart from that, you have something called commercial biotechnology products such as 3-tac have been useful for the treatment of the certain Fusarium penicillium species. It is also used in manufacturing of enzymes. Therefore, the correct answer is option D. Next question here. Modern detergents contain enzyme preparations of dash. See, when we talk about modern detergents, okay, they will contain alkali files. Okay, what are these alkali files? They are live in alkaline environments. Okay, they turn to become live in the alkaline environments like the soda or the alkaline soil, and the pH of the alkaline substances is about 9 to 11 on the pH scale. The alkali files keep on alkaline level of about pH 9 inside the cells, but the environment has a higher level of alkalinity. So alkali files can keep the right pH level by pumping in the hydrogen ions. That is, it will pump the hydrogen ions whenever it is necessary. Okay, so you have a lot of detergents which come with different enzymes. Based on that, the dirt from the clothes go. So now at this current era, if you see, modern detergents contain enzyme preparations of alkali files. The correct answer is option B. Next question here. Human insulin is being commercially produced from the transgenic species of dash. Very interesting question. I think by now you have guessed the answer. It's a very easy one because it's a common question that all of us know. It is none other than Serratia. It is simply abbreviated as E. coli. Human insulin is being commercially produced from the transgenic species of Escherichia coli. It is a bacterium and it is usually a gram-negative bacterium which takes in the pink stain during the gram staining process. The bacteria can also be grown easily in its genetics and comparatively simple. See, when compared to the other microorganisms, this is very easy for us to culture in the in vitro condition. Okay, and this is a prokaryotic model organism and it is important species in biotechnology where it has given a lot of benefits by culturing it. Therefore, the correct answer to this question is option A. Next question here. Probiotics are dash. Very easy question. We all intake probiotics in our food, right? So what are they actually? They are none other than but the live microbial food supplements, but they will not cause any harm to your body but rather cause benefits for your body. Probiotics are defined as some live microorganisms which include lactobacillus, and you have many species actually. Sometimes even certain species of yeast are also good to have, they say. They are beneficial for the host upon the ingestion of improving the balance of the intestinal microflora. So we all know when it comes to our intestine, we have some microorganisms which are innate. That is, right from birth we have certain microorganisms and they are beneficial for the intestine. They don't allow the other foreign materials from entering in, and in case if it enters, it's tried to eradicate out. So there are many beneficial functions from the intestinal microflora that we have. So This probiotics are actually recommended by many people to take it as food. Many yogurt come in that flavored. So all this is good for the intestinal microflora. Therefore, the correct answer to this question is option C. Next question here. Which one of the following pairs is wrongly matched? So we just need to match and we'll identify. So yeast is ethanol, correct. Streptomyces is, it's antibiotic, that is also right. Coliforms, vinegar, this is where we go wrong. Methanogens, cobar gas is also correct. Coliforms are a broad class of bacteria. Okay. They are bacteria in fact, and they are formed in our environment, including the feces. Say mostly coliforms are from the production of the feces from the humans and other warm-blooded animals. The presence of coliform bacteria in drinking water may indicate a possible presence of harmful diseases. So coliforms is not used in the production of vinegar. They are actually harmful when intaken, when they are ingested as in the water. So when we intake the contaminated water, they cause many intestinal problems, and sometimes if we leave it untreated, it might also become serious. The one which is wrongly matched out of the 4 options is option C, coliforms. It is a bacteria and you don't have any boon from it. Okay, next question here. Which one of the following pairs is not correctly matched? Again, it's an easy question for you. Streptomyces, we just now saw it is antibiotic, that is right. Serratia, drug addiction, wrong. Spirulina, single cell protein, that is also right. Rhizobium, biofertilizer, that is also right. Now to talk about this one, what we have, Serratia. Do you know what is it? Any idea? We have many species which come under this. Now when to talk about Serratia, Serratia marsins is a species of rod-shaped gram-negative bacteria, and it is also used as a facultative anaerobic organism, classified as an opportunistic pathogen in many cases. So it does not involve in drug addiction and all, but it's pathogenic. Okay, it is considered as a human harmful human pathogen which can cause, so it is related with urinary tract infections. So far that is what we have got information. Serratia is normally not harmful to healthy people, but it is known to cause some opportunistic pathogen or opportunistic infections in humans. Therefore the wrong one, which is correctly, which one is not correctly matched here, is option B. Okay, it is not involved in drug addiction. Next question here. Dough kept overnight in warm weather becomes soft and spongy because of dash. Okay, this is very easy question. You must have seen your mom making batter, dosa batter and all that in home. They used to mix it with hand, add some salt, and they keep it overnight in the outside. They don't keep it in the fridge. What is that process actually? And when you take it the next day, you see that the batter would have become fluffy and it will be little raised. You must have seen this. So what process is that? Very easy question. Yes, it is fermentation. Cohesion or the force is a force. Now we'll discuss one by hand. Cohesion is a force of attraction between similar molecules, while osmosis is a movement of solvent of molecules from a region of lower solute to higher solute concentration. But when we talk about fluffiness, softness, sponginess, the raising of the dough and all that is because of the fermentation. Okay, therefore the correct answer is option B. Next question here. The term antibiotic was first used by dash. Okay, this is again an easy question for you. Okay, this is a direct question. You just need to know who termed it actually. It is none other than but option C, Waxman. The term antibiotics was coined by Waxman, but it was first known antibiotic was penicillin, which was extracted from the fungi Penicillin notatum by Alexander Fleming in 1944. Antibiotics are chemicals. What do they do actually? They are chemicals that are produced from living organisms used to kill the other living organisms. About 2,300 antibiotics have been discovered so far. For example, you have streptomycin, tetracycline, and so many other things. So who termed the word antibiotic? It is option C, Waxman. But the first antibiotic was found by who? Alexander Fleming in 1944. It was from the fungi Penicillin notatum. Okay, next question here. During the formation of bread, it becomes porous due to the release of carbon dioxide by the action of, it's none other than yeast. See, these questions are very simple questions because the ones that we see in our day-to-day life. Yeast carry out fermentation, as we all know. That is why the other name of yeast is also called as baker's yeast. We all know that, right? And the one that we use for alcoholic fermentation, we call them as alcoholic yeast, or we call it as brewer's yeast. They release carbon dioxide, making the bread soft and porous. Hence, Saccharomyces is referred to as baker's yeast. Okay? Fine. Next question here. Industrial production of ethanol from starch is brought about by, again, this is a direct question for you. By now, you know what the answer is. Yes, it is Saccharomyces. Yeast helps in ethyl alcohol formation, while lactobacillus is involved in the milk bacteria. And penicillin is a source of penicillin. It's a fungi. Okay, therefore the correct answer is option A. Next question here. Which one of the following microorganisms is used for the production of citric acid in industries? We all know, we already discussed in the part 1, that fungi are majorly used for the production of various acids like malic, oxalic acid, citric acid. And we also know what the answer is. If you had gone through the part 1, now you will know what the answer is. Same. Out of 4, the correct one is Aspergillus niger. It is known as a guinea pig of the kingdom fungi. The main source of commercial preparation of citric acid is the fermentation of sugar with Aspergillus niger. So why is it called, or why is it termed as a guinea pig? Have you ever noted that? See, when people started researching, they used a lot of guinea pigs for their research. So guinea pig, in the sense itself, it is research. So Aspergillus niger is the guinea pig of the fungi, because a lot of research and a lot of experiments are carried out with this particular fungi. That is why it is termed as a guinea pig of the kingdom fungi. Therefore the correct answer is option B. Next question here. Yeast Saccharomyces cerevisiae is used in the industrial production of dash. It is ethanol. Yes. So here you have an equation here. Maybe you need to learn that. So glucose, the formula, the molecular formula is C6H12O6. In the presence of zymase and yeast, it produces ethyl alcohol and liberates carbon dioxide. So yeast, as we all know, it performs alcoholic fermentation. That is why you have another name for it as brewer's yeast. Alcoholic fermentations, they have ethanol in it, and that is why, see, ethanol is not good for consumption. That is why we say do not be alcoholic. Now, the correct answer to this question is option C. Next question here. Which one thing is not true about antibiotics? So we need to identify the wrong one. The term antibiotic was coined by Waxman. That is right. First antibiotic discovered was Alexander Fleming. That is also right. Each antibiotic is effective only on one particular kind of germ. That is where we go wrong. Each antibiotic is effective only against one particular kind of germ is incorrect. Okay, some persons can be allergic to a particular antibiotic. That is also correct. See, when we talk about each antibiotic, they don't go as a target thing. When we incorporate, there might be a response from the antibiotic side, or there might not be response, because we cannot target any organisms when we give an antibiotics. Now when you have fever, you are taking paracetamol. Likewise, they given a general medication, and only when the disease doesn't resolve, they go in for target. So each antibiotic is effective only against one particular kind of germ is not accepted. Okay. Next question here. The organism that is used for alcoholic fermentation. It's again a repetitive question here. Yes, the correct answer, as you all know, is option D, Saccharomyces. Okay. So the ethyl alcohol and carbon dioxide that are formed as the end process. Ethyl alcohol will be the end product, and carbon dioxide will be the liberated gas during the process. Okay. Next question here. Which of the following is wrongly matched here? Trichoderma has cyclosporin. That is right. Monacis has statins. That is also right. Streptococcus produces streptokinase. That is also right. Now the last one is wrong. Clostridium buticulum does not produce lipase. It is used for the butyric acid production. So this is where we go wrong. Option D is wrong. It does not produce lipase. Instead, it produces butyric acid. Therefore, the correct answer is option D. Out of the four, which one is wrong? If you say, it is option D. I again repeat, Clostridium buticulum actually is used in the production of butyric acid. Okay, therefore the correct answer is option D. So with this, we complete the part 2 of microbes in human welfare. Hope to meet you in the next part of the same chapter. Thank you.